day, dear students of dentistry faculty. Today we will continue with you the lectures on digestive system. Today you will listen the third lecture on this system. I am Əyyubova, Senior Lecturer of Department of Histology, Embryology and Cytology of Azerbaijan Medical University. Today we will focus on general morphofunctional features of glands of the digestive system. They are salivary glands, liver, and pancreas. First of all, the salivary glands. All of these collectively glands of the alimentary canal, which are associated with alimentary canal, they have very important functions and they aid digestion. They open into the alimentary canal via their ducts, and they release their secret via their ducts into this canal. First of all, let's talk about the salivary glands. The salivary glands, these are paired glands which are associated with the oral cavity, and they are paired parotid gland, submandibular and sublingual glands. All of them they have the same principle of structural organization. All of them are covered by connective tissue capsule. From the connective tissue capsule arise septa number 2. This septa will subdivide the gland into individual lobes and lobules. Here we can see one lobule, then the next lobule, and the third one. Inside of this connective tissue, we will have blood vessels, nerves, and lymphatic vessels, which are needed to supply the parenchymatous elements of the gland. And if we will look at their general morphofunctional features, we have to know that if it is exocrine gland, so all of them have secretory portion and the ducts. Their ducts are very complex systems. We will talk about them later. Now let's look at the schematic representation of their general morphofunctional features. Their secretory portions, pay attention, they are secretory portions, and these are system of ducts. Here you can see also the secretory portions and ducts. Secretory portions may have shape of acini or alveoli or tubules. In secretory portions, we can distinguish three kinds of cells. First of all, that are the serous cells. Here you can see. Serous cells, these are protein-producing cells, but they produce in low amount also carbohydrates. Because of this protein-producing cells, so they reach in rough endoplasmic reticulum, in ribosomes. In apical portion, you will see secretory granules, which contain the enzymes. What is the enzyme which is produced here? That is amylase, salivary amylase, or ptyalin. In small amount, also produced salivary lipase. Also, inside of the saliva, we will have lactoferrin, lysozyme, thiocyanate. These are antibacterial substances. Also, these cells will produce secretory component of immunoglobulins A. These are antibodies. So inside of saliva, we have not just enzymes, we also will have these antibacterial substances, which will control microbiota in our oral cavity. And in lateral sides, we will see tight junctions, so the cells attached to each other via tight junctions. Above region of tight junctions, we can see intercellular canaliculi. They open into the lumen of the secretory portion. And below tight junctional regions, we can see interdigitations. These interdigitations were located between the neighbor cells. And then next type of the cell here. Next type of the cell, these are mucous cells. Pay attention. These are pink colored cells here. And in this one, these are white colored cells. The serous cells, by the way, these are blue-colored serous cells, which have blue color. Actually, they, by hematoxylin eosin staining, their cytoplasm will be colored into the blue color because they are rich in RNA. That's why you know that is basophilia. They will attract basic stains, that is hematoxylin. Mucous cells, they are larger than serous cells, and inside of their general structure almost the same. Their shape, they are truncated pyramid, shape of truncated pyramid, and but the nuclei has flattened shape and located at the basal region. If we will look at the nucleus of this cell, pay attention, of the serous cell, they are rounded. But in mucous cells, that are nuclei have flattened shape and located at the basal region of the cell. In apical region, we can see so much large secretory vesicles that contain mucinogen. Mucinogen, then when it is released into the lumen, then it will release into the oral cavity. It will bind water and will be converted into mucin. But inside of the oral cavity, the mucin will be mixed with the content of the oral cavity. By this way will be formed mucus. So these are mucus production cells. Then we have here myoepithelial cells. Myoepithelial cells, they have processes, and these processes, they surround the secretory portions. These secretory, pay attention, these are these cells. They are myoepithelial cells, and here they have the red color. They surround secretory portions and they share basal lamina of these secretory cells. Inside of these myoepithelial cells, we can find actin and myosin proteins. It means that they have ability for contraction. When the processes contract, these are, can you see these processes? When they contract, they press on the secretory portion, by that way facilitating release of the secrete into the lumen of the secretory portion. Then we have the ducts. The duct system of salivary glands, this is something compound, complex. It starts as intercalated ducts. These are intercalated ducts, and they continue into striated ducts. Intercalated ducts surrounded by cuboidal cells, cuboidal shaped cells here. That is illustration from your album, by the way, and so that you will know when you will work at your albums. So that is cuboidal shaped cells which cover the striated duct. Inside of these cells, so these are cells which that is simple cuboidal epithelium. Around them we also can find myoepithelial cells. Pay attention, here we can also find these myoepithelial cells. Intercalated ducts will continue into striated ducts. Pay attention here, striated ducts. Striated ducts covered by simple columnar epithelium. And in the basal cell membrane, we can see so much invaginations. These invaginations, these are folds, enfoldings. They subdivide the basal compartment of the cell into individual longitudinal compartments. Inside of these compartments are located mitochondria. These mitochondria needed for providing energy for the operation of sodium-potassium pumps, which are also located in basal cell membrane of these cells. This sodium-potassium pump will carry out sodium from the content of the lumen of this duct. By that way, it will lead to absorption of these ions, and that is very important in the production of the secrete. And okay, then let's go to here. You can see the striated duct cell, the mitochondria, and enfolding. Striated duct system, they fuse with each other and form increasing caliber of the duct system. That will be interlobular, then intra and interlobar ducts. All of them together will fuse with each other and they will form terminal duct of the gland, which opens into the oral cavity. Here we also have to know that the product of these glands, which is produced inside of the secretor portions, that is isotonic with the plasma of blood. But when this saliva flows into the duct system, after the intercalated, we know that they are striated ducts. In striated ducts, that saliva is modified by the activity of these cells. They will take up sodium, then chloride ions from the lumen. They will carry these ions into the connective tissue surrounding and then into the blood capillaries. And hydrocarbonate anions together with potassium ions will be released into the saliva. Then the water also will be absorbed. So finally, the final saliva will be hypotonic. The saliva is needed for lubricating the surface of oral cavity, then moistening the food. It will help in swallowing, formation of the bolus, and also the saliva has antibacterial activity. And saliva also needed for taste discrimination because food products will be dissolved here. Then we will look at the individual glands. First of all, parotid gland. Parotid gland, it is mainly serous gland. It constitutes approximately 30% of all salivary output. Then here we are looking at the histological section of the parotid gland, and here we can distinguish the secretor portions, which mainly constitute the gland that are acinar secretor portions, which include mainly serous cells, which produces proteinous secrete. Here we are looking at the cross sections of these secretor portions. Then intercalated ducts, pay attention, they covered by cuboidal epithelium. Then we are coming to striated duct, which composed of the columnar epithelium, which striated both the striated region of the basal part of the cell. Then we have the interlobular duct. It is covered by 2 or 3 layers of cuboidal cells. So with increasing caliber of the ducts, the layers in the wall of these ducts will be increased. So final duct will be covered by stratified squamous non-keratinized epithelium. Now we are coming to mixed gland, that is submandibular gland. Submandibular gland will include 3 kinds of secretor portions. First of all, acinar secretor portions, which composed of mainly serous cells. Then we will have mucous secretor portions, which include mainly mucous cells, which have light appearance. Then we have mixed secretor portions. Pay attention to this secretor portion. In the lumen of the end piece, we will see, or secretor portion, we will see mainly mucous cells, but at the periphery of them we will see serous demilunes, which located at the periphery. They surround like half moons. They include serous cells. That is the mixed secretor portion. If we are talking about the submandibular gland, its output constitutes 60% of all saliva. And the 90% of its output of its secrete mainly include serous secrete, but 10% there will be also mucus. So that's why the main secrete of the submandibular gland is serous component. And the duct portions we can see also here, intercalated ducts. If we will look at this section of the submandibular gland, we can clearly see here the mucous cells, serous demillions as half moons, and mucous cells also. Pay attention to this section also, mucous cells and intercalated striated ducts here. Now we are coming to sublingual gland. Its output constitutes 5% of all saliva output. And sublingual gland, if submandibular gland has its own terminal duct which open into the oral cavity at its bottom, the sublingual gland don't have its terminal duct. It has several ducts which may open individually into the bottom of the oral cavity, or they open into the duct of the submandibular gland. Now let's look here. These are secretory portions, cross sections of secretory portions. As you can see here, the cells have light cytoplasm, so they are mucous cells. Here also we can see them. Then striated ducts, intercalated ducts, striated ducts, so the duct system which is present in connective tissue septum that separate the lobules of the gland. Inside of the septa, you know, the travel blood vessels, nerve endings. Now we are coming to the largest gland of the human body, that is the liver. You know that liver is situated in right upper quadrant of the abdominal cavity, just under the diaphragm. And you know that liver includes 4 lobes, right, left, caudate, and quadrate. It is covered by Glisson's capsule that is invested by serosa. Then, at the inner surface, we have hilum, that is indentation of that part of the organ. The liver, 1.5 kilogram its weight. Liver, it is laboratory of the body. It performs more than 100 functions. Liver receives blood from 2 sources, that is hepatic artery, that is branch from abdominal aorta, and portal vein, which carry blood from the unpaired organs of the abdominal cavity. Portal vein is formed by a fusion of 3 veins, as you know, that is splenic vein, which carry blood from the spleen, and it rich in hemoglobin components, bilirubin, and so on. Then superior mesenteric and inferior mesenteric veins, which carry blood from the intestines, mainly that is the blood which rich in nutrients. After entrance into the organ, these 2 blood vessels will branch together. Now let's look at the internal structure of the liver. In histological section, the structural and functional unit of the liver, these are liver lobules. They have shape of hexagonal prisms. We have to know that connective tissue which surround the organ, this Glisson's capsule, then at the hilum, it forms thickening, and through that region, the connective tissue enter the organ. And here we will see how the connective tissue will branch, and it will subdivide the organ into lobules. Here we are looking at the liver of pig, and that is the liver of human. What is the difference? As we can see, the boundaries between the lobules are exact, and in liver of pig we can distinguish individual lobules from each other, but in human liver we have to just estimate the boundaries of individual lobules because of the scarcity of the connective tissue elements inside of the liver of human. In the center of the lobule, we can see central veins. At the boundaries between when the 3 lobules meet each other, here we will see portal areas. These portal areas will contain blood vessels, also nerves. Inside of these portal areas, we can see 3 kinds of main structures. That is the interlobular bile duct, interlobular artery, and interlobular vein. These are branches of large vessels, of course. They located in portal areas. Main cellular element of the liver, these are hepatocytes. These are main cells which form parenchyma of the organ. But if we are talking about the portal areas here, the connective tissue of the portal area separated from the hepatocytes by the layer of modified hepatocytes, which called limiting plate, that they form layer here. Between limiting plate and this connective tissue elements located space, which is called space of mole. It is here, here will be collected the lymph from this organ. You have to know that the 50% of lymph which is formed inside of the body, that is produced inside of the liver. So here, that is the first region where the lymphatic vessels will arise and they will carry out lymph from this organ. Let's talk about the blood supply of the liver. As I told you, the liver receives blood from two sources, that is hepatic artery and portal vein. These two vessels will branch inside of the organ together. They will go together. In the hilum, they will branch into right and left branches. Then they will, inside of the organ, they will subdivide into segmental branches. Finally, they will come at the boundaries between the lobules in the portal areas. They will form interlobular vein. If the branch of the portal vein, it will be interlobular vein. If it is branch of hepatic artery, it will form interlobular artery. And as we know, in portal areas, we have also other component, that is interlobular bile duct. So three structures we will see here, they form triad here. Then here in the center, we can see classical liver lobule in the shape of hexagonal prism. At the periphery of this lobule, we will see that from this artery and vein arise small branches. They called inlet arteriole and inlet venule if in the case of the vein. Then these two vessels will fuse at the periphery of the lobule. They will fuse with each other and they will continue into sinusoidal capillaries. These sinusoidal capillaries will come to the center. They will radiate towards the center of the lobule, and in the center we will see central vein. So they will carry blood into the central vein. From the central vein, blood will flow into the sublobular veins. The sublobular veins will come together and they will form collecting veins, which will finally form the right and left hepatic veins, which will carry blood into the vena cava inferior. By the way, the hepatic artery, it is a branch of abdominal aorta, which carry here highly oxygenated blood, and it constitutes 25% of the blood. But 75% of the blood comes from the portal vein. Now, let's look at if we will talk about the blood which flows within the sinusoidal capillaries, that is a mixed blood because it comes from both sources, from arteriole and from the venule. That's why in sinusoids will flow mixed blood. Between the sinusoids, we will see plates of hepatocytes which also radiate towards the center. Each this plate will contain in young persons there will be 2 rows of hepatocytes. In more adult persons we will see just 1 row of hepatocytes. Then let's look at these plates. Pay attention. So as I told you, plate include 2 rows which form plates. That is one plate, that is other one. Inside of these plates, between the lateral surfaces of hepatocytes, we will see green-colored bile canaliculi. These bile canaliculi, pay attention, they will go to the periphery, run to the periphery of the lobule, and they will join interlobular bile duct. We have to know that if the blood flows from the periphery of the lobule towards the center, then the bile will flow in opposite direction from the center of the lobule toward its periphery. Then we will talk how these lobules are organized. Here we can see the plates of hepatocytes. Hepatocytes also have shape of hexagonal, these are polygonal shaped cells. Pay attention to one of these cells. In the center we can see nucleus. Because hepatocytes are metabolically active cells, they not just produce bile, they also produce many the proteins of the plasma, and they participate in detoxification of toxic substances, drugs, and also hormones. So they have various different functions. That's why they reach in many organelles. First of all, mitochondria, which will provide energy for its functioning. Then Golgi apparatus, endoplasmic reticulum, rough for production of proteins, and smooth for detoxification of toxic substances. Also, they include peroxisomes. Then on that surface, we will see that each hepatocyte will have lateral surfaces. Pay attention, that is lateral surface. These are lateral surfaces. Then a sinusoidal face here, which faces sinusoid, and biliar face, which faces the bile ductal. Pay attention, or bile canaliculi. Bile canaliculi located between the lateral surfaces of the hepatocytes, and here the cell hepatocyte will release its bile. At the bile canaliculi, that's at the region of it, we will see tight junctions which isolate intercellular space and protect by that way the intercellular space from entrance of the bile. Between the rows of hepatocytes, we can see sinusoid. Inside of the sinusoid flows blood. As we know, that is mixed blood. Pay attention that these sinusoids, as we know, they are vessels, capillaries with large diameter. They lined by sinusoidal cells. These are endothelial cells. Pay attention. These are elongated endothelial cells. Between these endothelial cells, we will have gaps, fenestra. Then these endothelial cells do not have their basal lamina, but they are surrounded by reticular cells, are surrounded by the reticular fibers. These are collagen third type. They present here in this space. This is the space which is located between the hepatocytes and the wall of the sinusoids. So in sinusoidal lining cells, these are endothelial cells separated from the hepatocytes by the space of this. Here we can see this space. In this space we can see microvilli of hepatocytes which are located here, and they will increase contact with the plasma that escapes from the wall of the sinusoidal capillary. Because of the fenestrations, it is free way for the plasma components into the space of this, and in this space the plasma components come into close contact with the hepatocytes, and the hepatocytes will exchange here the substances they will take from here, necessary for example nutrients, and the hepatocytes will do their functioning here. Also, we have to know that these reticular fibers are produced by some cells, ethos cells, which also present in this space between hepatocytes. These ethos cells, these are fat-storing cells. They store vitamin A. This is fat-soluble vitamin. Also, ethos cells are responsible for formation of the scar tissue during damage of the liver. They will be activated by tumor growth factor, which is produced by the hepatocytes. when the damage, when an insult into the liver, the these little cells will be converted into myofibroblasts, then into the fibroblast cells. Then inside of the liver, we also will find the pit cells. These are actually natural killer cells of the liver. Then let's look at the next illustration. Here I would like to tell you about something about the bile canaliculi. The bile canaliculi, which start in approximately at the center of the lobule, and they continue into the interlobular bile ducts. When this and we know that these are bile ductules, canaliculi, they do not have their own wall. Their wall is composed of the hepatocytes. Near to the junction, near to the passage of this bile canaliculi into the portal space, here they will continue into small canals, which are called canals of Hering. And in the wall of the canal of Hering, we will see hepatocytes, and besides hepatocytes will be located ovoid cells. And these ovoid cells will cholangiocytes. They will also cover, then we will find them in the wall of the interlobular bile duct also. And these cells are regenerative cells. They will regenerate and replace hepatocytes when it will be needed. So here we can see pay attention to this illustration. Now we are looking at the two hepatocytes, between them bile canaliculi. Then this space, pay attention. Then endothelial cells of the sinusoid. So here we will see one sinusoid, in other side, other sinusoid. And pay attention to one point, key point here, that hepatocyte, one cell, in one side it faces blood, in other side it faces bile, but these two fluids never mix with each other. If they will mix, that is parenchymatous hepatitis, and that is big problem. That is a pathological condition. And ethos cells we are looking here. Then here we can see the Kupffer cells in the wall of the sinusoid. Kupffer cells, these are macrophages of the liver. They are resident antigen-presenting cells of the liver. They will phagocytose microorganisms, and they will phagocytose cellular debris and defunct erythrocytes. So that is the main function, that is phagocytosing. Then pay attention to next illustration. That is central vein in the center of the lobule. Here we can see plates of hepatocytes, between them sinusoids located. Then here the portal space. I want to show you the portal space here. It is surrounded by connective tissue elements, and here we can see the interlobular bile duct, then interlobular artery, vein. These are components of portal space. But pay attention to the lymph vessel which arises here. And we also know that here the hepatocytes, here present hepatocytes, and also, so that is the connective tissue elements of the portal space. Here we are looking at the silver impregnation of the liver, which will show us reticular elements, the reticular fibers of the liver. We have to know that besides classical liver lobules, there are other conceptualizations about the liver structure, which is proposed by histologists. Why we have additional conceptualizations? First of all, let's look at this illustration. That is the classical liver lobules. These are hexagonal prism-shaped lobules. Now we know their structure. Now let's look at this structure, that is portal lobule. That conceptualization is acquired because of the, if we will talk about the liver as a gland, because as we know, in other glands, in the acini of the glands, the product of the gland will flow from the periphery towards the center of the gland. But in this case, in the classical liver lobule, the bile, as we know, flows from the center to the periphery, and it is not in accordance with conceptualization of the gland. That's why histologists proposed other lobule, that is portal lobule. It has triangular shape. In the center of it, we will have an interlobular bile duct, and these regions and the bile which produced in these lobules will flow into this interlobular bile duct, which is located in the center. So that will be portal lobule according to the functioning of the liver as a gland. In this case, the bile will flow from the periphery of the portal lobule towards the center, towards this interlobular bile duct. And that is other conceptualization. It is liver acinus, which has diamond shape. Pay attention to this illustration. This diamond shape, it has this angels, and these angels located central veins, and these angels located portal areas. That is according to the blood supply and how the rate of degeneration of hepatocytes during insult, during toxic and toxic situations, and also hypoxic situations. Let's look here. We know that around the liver lobules located distributing arterioles and venules, and from them they are distributing arteries and veins located around. So in this region we will have distributing arteriole. From it will arise inlet arterioles, as we know, that enter into the lobule. So the first region will receive more oxygenated blood, most oxygenated blood. Second region will receive less oxygenated blood, and the third region, which is far away from the source of arterial blood, it will receive the least oxygenated blood. That's why in pathological conditions, if it will be hypoxia, on that case the first region which will be suffered from deficiency of oxygen, it will be the third region. So that is according, we will talk about if we are talking about the pathological conditions of the liver. Now let's look at something about the functioning. As we know, the liver cells perform so much functions. They participate in metabolism of proteins, they produce proteins of plasma, then proteins of their own use. Here we are looking how the proteins, amino acids are taken up by hepatocytes, proteins are produced, then transported to the Golgi apparatus, then so the proteins of the plasma, albumins, fibrinogens, and most of the globulins also, exception gamma globulins. They released into the blood circulation. Then it participates in the metabolism of carbohydrates. It takes up glucose, produces glycogen. Then when it is needed, for example, during fasting, glycogen is broken down if it is needed, and needed glucose will enter the blood circulation. Also, hepatocytes participates in lipid metabolism. It produces lipoproteins, and so on. Another function of the liver is detoxification, inactivation of hormones, barbiturates, antibiotics, and storage also. Another function, storage of vitamins, lipid soluble vitamins, mainly vitamin A in scarce amount, vitamin D and K in very low amount, vitamin B12 also. So that are the functions of the liver. Now let's come to the pancreas. As we know, the pancreas here, it has uncinate process, then it has head, then body and tail. Pancreas, it is a gland which has exocrine and endocrine functions. Exocrine function, it is related to the production of the digestive enzymes, and endocrine, it is the production of hormones. Let's look at the section of the pancreas here. That is histological section which is stained by hematoxylin eosin staining. The secretory portions constitute compound tubular alveolar gland, which include, pay attention, secretory portions and duct systems. If we are talking about the secretory portions, so the unit of the excretory portion of the pancreas, that is called acynus. The acynus, it has sac-like structure. Here located secretory cells which have shape of truncated pyramid. And then here we can see central acinar cells which have green color. Central acinar cells, that is beginning of the duct system, and it continue into the intercalated duct. Presence of central acinar cells, that is characteristic feature of the pancreas. If we are talking about the cells, secretory cells, these are they have features of protein-producing cells. In the basal part, we have nucleus, then rough endoplasmic reticulum, then ribosomes. That's why the basal region, pay attention to the slide here. Basal region have blue color. It has basophilic features. And the apical part of the cell, apical portion, it rich in zymogenic granules. That's why this region is called zymogenic region. And it has acidophilic cytoplasm because of so much granules, which include pancreatic proenzymes, not activated but proenzymes. Exocrine pancreas produces pancreatic juice, and it is activated during meal ingestion. By neuronal and hormonal activation. These secretory cells has receptors for acetylcholine, which is a neurotransmitter that is released from parasympathetic nerve endings. Also, they have receptors for cholecystokinin, that is a hormone which is produced by DNES cells, diffuse neuroendocrine cells, which are located in the wall of the alimentary canal. And but these central acinar cells, they have receptors for the other substance, other hormone, that is secretin, and also they also have receptors for acetylcholine. They are activated by these components. If these secretory cells produce enzyme-rich fluid, then central acinar cells will produce bicarbonate-rich fluid, and it will be produced in response to those hormones. So pancreas will produce, exocrine pancreas will produce two types of fluids. First, enzyme-rich fluid. Second one, alkaline bicarbonate-rich fluid. That alkaline fluid is needed for neutralization of the acid environment of the chyme, which is coming from the stomach into the duodenum. As we know, the pancreatic duct will join the common bile duct before opening at the duodenum. Now let's look here the central acinar cells, that is secretory portion acynus, and they are acinar cells. Intercalated ducts we are looking here, and the blood capillaries which surround the secretory portions. Then after the intercalated ducts, we will have intralobular ducts which will continue into the interlobular ducts, and they will join each other and form pancreatic duct. So we cannot see here striated duct which we saw in the case of salivary glands. So here these intercalated ducts and the wall of these cells will also have sodium-potassium pump, which will carry sodium and it will also a little modify the content of the pancreatic juice. The enzymes which are released from these cells, pay attention to electron micrograph representation of the secretor portion. Here we are looking at these cells. They produce proenzymes. That is trypsinogen, then it will be chymotrypsinogen, then and elastase, desoxyribonuclease, ribonuclease, lipase, and other enzymes, but they are actually non-active when they are released from these cells. Also, these cells will produce trypsin inhibitor, which will prevent activation of the enzyme inside of these cells. Activation of the pancreatic enzymes will take place just in the lumen of the duodenum. Then we are coming to endocrine portion of the pancreas, that is islet of Langerhans, which is located inside of the gland between the exocrine portions, and these islets are surrounded by a network of reticular fibers. Inside of these islets, we can distinguish several types of cells, but most of them, these are A cells, B cells, and D cells. These cells will produce their own hormones into the blood circulation. Mostly cytoplasmic staining with Mallory-Azan stain. A cells will have a red color, B cells brownish orange color, D cells will have blue color. A cells produce glycagon. The glycagon, that is hormone which will increase level of glucose in blood. The glycagon produced in non-active pro-glycagon state. Then in Golgi apparatus, it will be cleaved and the active hormone is released from the cell. B cells produce insulin and they constitute 60-70% of all cells. Insulin, that is hormone which decreases level of the glucose in blood, and the insulin, it has receptors on the surface of our cells, the muscle cells, then liver cells, and other cells of our tissues. And in response to insulin, in our cells will be released transporters for glucose transporters, which are abbreviated by GLUT4. These are transporters of glucose. They are stored under the cell membrane, and they will be released into they will incorporate it into the cell membrane and in response to insulin. They will increase absorption of glucose from the blood circulation into the cells, into our tissues. The level of insulin increases at some conditions. It is increase in the level of glucose, amino acids, lipids, also increase in the level of stress hormone. So the stress hormone, cortisol, also increases the level of insulin in blood circulation. Obesity, all of these factors, growth factors, then hormones which released from diffuse neuroendocrine cells in the wall of the alimentary canal. The factors which decrease level of insulin, they are fasting, drop of the glucose in the level inside of the blood circulation, also somatostatin and leptin. The other hormones we have to know here, these cells produce somatostatin. Somatostatin will inhibit functioning of all of the alimentary canal, its motility, functioning of glands, functioning of parietal cells also in the wall of the stomach. Then we will have other cells which located in less amount in the wall of the pancreas. They are the cells which are not shown here. They are G cells. They produce gastrin, which induces, activates and stimulates activity of the stomach. Also, we have here P cells which produce pancreatic polypeptide, D1 cells producing vasoactive intestinal peptide. Then we have epsilon cells which produce ghrelin, activating the smooth muscle relaxation peristaltic activity of the stomach, and it induces the feeling of hunger. EC cells produces substance P. These are the cells which we can find inside of the islets of Langerhans endocrine portion of pancreas. A little thing amount, let's look at this illustration, how the insulin activates our cells. It's insulin receptor on our cells of the tissues. It will activate the glucose transporters, which is needed for carrying of glucose inside of the cells. Let's go to gallbladder. Gallbladder, you know, that is sac-like organ which is located under the liver, and it is needed for accumulation and the concentration of bile. Gallbladder, it has the body and it has neck. The neck continues into the cystic duct. At the continuation of the neck, we have some outpochic, like pocket-like outgrowths, where will be accumulated gallstones. About the histological structure of the gallbladder, we can tell that here we can distinguish three layers, that is mucosa, muscularis externa, and the adventitia or serosa. If we will look at the internal surface of the gallbladder, we can see here folds, which increase surface area of this organ, and it is formed by the lamina propria evaginations which enter the epithelium. It will formed by the lamina propria. The epithelium which covers the gallbladder, simple columnar epithelium. Here the cells have microvilli on the surface, and here we can distinguish two kinds of cells. These are dark cells and clear cells. Then lamina propria, it is loose connective tissue rich in elastic fibers and blood vessels. Then we are coming to the muscularis externa, which include one layer of spirally oriented muscles, smooth muscles, pay attention. And at the neck of the gallbladder, in lamina propria, we can find tubular alveolar glands, which produce mucus for lubrication, the surface of the gallbladder. Outermost coat will be adventitia, on the face of the gallbladder that faces the liver, and on the inferior surface of the gallbladder will be covered by peritoneum. That's why that coat will be serosa. And finally, the cystic duct, which arises from the gallbladder, will be fused with common hepatic duct, and they will together form common bile duct. And common bile duct together with pancreatic duct, they will fuse with each other and form ampulla of water. And at the papilla of the duodenum, major papilla, this duct will open into the duodenum. By that way, the bile, which is produced in the liver, and then the pancreatic juice will be released into the duodenum to aid digestion. So that is the end of the lecture, dear students. So we finished the lectures on digestive system. Be safe, take care, see you on next lectures.